Hello, welcome to another video from the conscientious biologist, Ben Gallagher. Now, this one's the detail of protein synthesis, and it's really, really important that you've just watched the video on the structure of DNA. That's the phase 2 structure of DNA video. It's the essential baseline information for now being able to access this details of protein synthesis. So don't watch this one until you've watched that one. It's only a very short video, but go back and watch it if you haven't watched it already. This is from the GCSE specification. However, this is probably the most significant part of the GCSE course in terms of pulling information down from A level. This video is literally within a whisper of A-level detail. It's only a few terms and a few little technicalities that are missing that would make this suitable for A-level specification. But remember, this is a GCSE level video, so if you are studying this at A-level detail, this hasn't quite got enough, but it's almost there. It's a really good one to watch if you're not sure, if you just want to recap some of your details of protein synthesis. As always, please do subscribe to my channel if you haven't done so already. Thank you. So back in phase 1, there were 3 lessons in a sequence. They're in a playlist called Proteins that look at protein synthesis. That gives you a really good overview and understanding of the process, which hopefully you've got. We're now going to pass some detail on top of that. But let's just recap that first. So the basic protein synthesis mechanism, and please remember, proteins do everything within a cell. Just put that picture up there as a little reminder to you that proteins are like robots working in a factory. The factory being the cell and the robots being the little proteins. And also remember, just as a visual reminder, that proteins are built of building blocks. The building blocks are amino acids. That's just there to try and remind you of the phase 1 stuff. But let's get rid of them for now. So let's say in the cytoplasm of a cell, you've got all these amino acids. They've got into the cytoplasm because you've eaten food, which contains proteins of other organisms. You've digested those proteins using protease enzymes. You've absorbed them through your small intestine. They've gone into your blood. They've gone to your cells. They've moved into your cells by diffusion. So your cells should now be full of useful amino acids. But you've got to build those amino acids into a useful protein for you. This is done, if you remember, by ribosomes. Ribosomes will read the instructions that are in the DNA. So if he's looking at this DNA above my head, he can see that the first one of those he needs is that kind of unhappy one. So he pulls that up. The one he needs after that is the sunglasses one. So he pulls that out of the cytoplasm, puts it in his chain. He then needs the one with the mustache. So he puts that in his chain. And then he just works his way down the list on the DNA to put all the amino acids in the correct order. Now that's the vital mechanism for protein synthesis. It's a ribosome reading the DNA, which is just a sequence of amino acids. The ribosome puts them in that correct sequence to make this, to make the polypeptide being the chain of amino acids, called a polypeptide because peptide is the name of the bond in between the amino acids. So between every one of those amino acids is a peptide bond linking it to the ones next to it. Okay, now this actually represents the first 2 stages of protein synthesis. In this video, we need to look at the details of those 2 stages. Hopefully you'll remember that there is a 3rd phase, which is the folding, because that specific sequence needs to fold into a very specific shape, that shape being determined by the sequence, and that shaped protein will have a specific function. Shape determines function. Okay. In this video, though, we're looking in detail at the first 2 stages. So let's have a look at those now. Okay. I think I said in the last video that this is one of the more complex things that you come across at GCSE. So really try and concentrate now. If you get through it and you're still unsure, watch it again, because it's a really, really vital bit of biology. It's really important for your understanding of so many areas of the course, including what we go on to from here, which are things like meiosis, genetic inheritance, genetic modification, all those kind of things. So really important thing. So let's look at stage 1 then. Stage 1 happens in the nucleus. And let's think about why. Because we're actually starting out with a problem. If you look at what we've got here in this cell, we know that the ribosomes need to read the genetic code which is written in the DNA. So I've just put a little yellow circle over there around a little bit of the DNA, because perhaps that's the little sequence of DNA, the gene, that codes for the protein that that cell currently needs to make. Now, you might notice there's a problem here. The problem is that the DNA is in the nucleus, whereas the ribosomes are out in the cytoplasm. Now, the DNA can't leave the nucleus. It's too big for a start. It can't go out through the nuclear membrane. But also, DNA is really precious. If it gets damaged, then the cell's not going to be able to make its proteins and the cell will die. So you need to keep it locked up in that nucleus. That's why cells evolved the nucleus. So DNA can't read the nucleus, but the ribosomes can't enter it. So they can't actually get to each other. And the ribosome doesn't have eyes. It can't peep its way into the nucleus. So you've got a problem here. But there's a solution. What you can do is you can make a disposable copy of the specific bit of DNA code, which is just the gene, the bit that I put in a circle up there, that one gene that's specific to the protein you need. And if you can make a disposable copy, that disposable copy can leave the nucleus and go to the ribosome. It's like making a photocopy that it doesn't matter if it gets damaged, because you've still got the original document that you made the photocopy from. Now, that new copy is called a messenger copy. Okay. If you're studying this at A level, you'll find out it's called mRNA, M for messenger, but you just need to know it as a messenger molecule. Okay, let's look at how that happens. So if I just expand on that section of DNA that I've marked in the circle, and I'm pulling up the DNA structure from the last presentation. That's why it's so important that you recognize what that is. So there's all my nucleotides with their different bases on them, the A, G, C, and the T. It's double-stranded look, but remember, it was that bottom strand that we put on there that's the coding strand. It doesn't have to be the bottom one. It depends which way around you flipped your DNA. But for mine, that one that you can read along, the GCTCAGG, blah, blah, that's the coding strand. That's the key information. So what needs to happen during stage 1 is that the DNA needs to open up. The word for that is that it unzips, just like a zipper on a jacket. It unzips because the bases peel apart from each other, and they actually look like the teeth that you have on a zip. An enzyme is going to come along, and it's going to read not the coding strand, not the strand that we want. It's going to read the other strand, and as it moves along it, it's going to make a complementary copy of that non-coding strand. Now, it should be pretty obvious to you why it's read that non-coding strand, because look at that copy that it's just made. Look at the black copy that I've put there. That copy, because it's complementary to the non-coding strand, is a copy of the coding strand. Compare the shapes. Look, the T on the end there is the same. The arrow pointed one, the C next to it is the scooped out one. It's an identical copy of the coding strand because it was made as a complementary copy of the non-coding strand. Hopefully that makes sense to you and you can see that they're the same. Now that we've got that, that can now move out of the nucleus and move into the cytoplasm. So if you look over on the cell picture I've drawn over there, obviously not to scale, but I've shown that molecule moving out of the nucleus, heading towards the ribosomes where they need it. Now the DNA doesn't stay unzipped, that just closes up and goes back to exactly as it was before. It's as if you've opened the book so that the enzyme could quickly make a copy, you've taken the copy away, and then you've closed the book up safely. That's stage 1, the creation of this messenger copy of the coding strand of DNA. Okay, let's move on to stage 2 now. So now we've got that messenger copy and it's moved out into the cytoplasm, and we know that once it's in the cytoplasm, a ribosome can associate with it, a ribosome can link onto it, and the ribosome reads 3 bases. We talked about that in the previous video on the structure of DNA, that it's 3 bases that code for an amino acid. So that ribosome is going to read those 3 bases. This is where something a bit odd happens. You get a specific transfer molecule that brings it a specific amino acid. So this is a whole new player that we've never talked about now. This transfer molecule is another little chemical that recognizes the 3 bases on the messenger molecule, so the big black one, And if you look even at how I've drawn it on there, you can see that my transfer molecule has got his 2 feet and his arms on the three bases. It's checking them out and it's going, yeah, okay, oh, those 3, they correspond to me. That transfer molecule knows its name in terms of the 3 bases that are there. And that transfer molecule will always, always, always carry the same specific amino acid. Okay, so in this case, if those 3 bases were ATG, then that transfer molecule would recognize those and go, oh yeah, that's me, I carry a methionine, that's great, I'm the methionine one that matches up to these. If it was a transfer molecule that came along, put its sort of hands and feet on the 3 bases of the messenger molecule and go, oh no, that's not me, it just disappears off. It's only when the correct transfer one with the correct amino acid on its back comes along and goes, oh, that's me. When it's there, it goes, brilliant, I'm the one carrying the amino acid that this code says it wants. So once it's done that, it gives its amino acid to the ribosome and it goes off and finds another amino acid. Not just any amino acid, of course, it will always find that same specific amino acid. If that transfer molecule wants a methionine and it's donated a methionine, once it's given it to the ribosome, it will go off and find another methionine amino acid. Okay, always carries the same one. Now what happens? The ribosome's got its first amino acid, so it can move down onto the next triplet, the next 3 bases. A new transfer molecule bringing the amino acid that corresponds to those 3 bases can come along, and then it'll head off and find a replacement for the one it's just donated. And then the ribosome moves down to the next 3. You should be seeing what's going on now. Another transfer molecule that matches the next 3 bases comes along with its specific amino acid and gives it along. Then it goes off and finds another one and the ribosome moves along again. You're seeing the pattern happen now, and it just repeats. The ribosome reading 3, recruiting the transfer molecule it needs, which gives it the correct amino acid, and then that one heads off and it moves to the next, and so on. Until we get to the last one on the chain, that last transfer molecule will bring the last amino acid. And of course, as it's been going along, as I've written here, it peptide bonds each amino acid in turn to the last one. And when it gets to the end, and that last transfer molecule heads off to find its new amino acid, the ribosome can come off the end of the mRNA, off the end of that messenger molecule, and you've created your polypeptide. Now, in reality, of course, your piece of messenger, your messenger molecule holding the DNA code would be way, way longer than just the 12 bases I've used here. It would be hundreds and hundreds of bases long, meaning your polypeptide would be much, much longer. Okay, if you had 300 bases of DNA, your polypeptide would be 100 amino acids long, because remember, it takes 3 bases per amino acid. So you'd create your polypeptide by the ribosome moving along, reading 3 bases at a time, with the transfer molecules bringing you the correct, specific, corresponding amino acid to that 3 base code. Really important you get that stage correct. So we're going to do 2 things now to end this video. We're going to put the 3 stages together as a diagram, and then we're going to do it in writing, in written bullet points, ready for you to learn and practice ahead of your exam. So let's put it all together. So using the diagrams that we had at the start of the video for the recap, we've got a ribosome, we've got the DNA, the ribosome reads the DNA to create this chain of amino acids that is your polypeptide. But let's put in our detail now. We know that the DNA doesn't actually look anything like that. Our DNA looks like that. It's our sequence of nucleotides with their specific bases as a double strand. We know that when that opens up, it can be read by an enzyme to create a complementary copy of the non-coding strand, which would be a copy of the coding strand. And that one's going to go to the ribosome. But of course, we know the ribosome isn't actually a little man like this. The ribosomes look like that. They're a kind of blob with these 2 subunits. They're going to read 3 bases at a time. And by doing that, and recruiting this transfer molecule to bring them the corresponding amino acid, they can create this polypeptide, this chain of amino acids. So those are our first two stages. Now remember, now that's got a fold, so let's just shrink that down to a much smaller line, and remember that actually it should be much longer. So we're going to represent it with this long blue line to represent the long polypeptide, the long chain of amino acids. So stage 3 is that long chain needs to fold in a very specific way, and it will fold depending on the sequence of amino acids that are there. Okay, when we draw it, we never draw it as that folded squiggly line, of course, we just draw around the outside of it so you can see the shape that it folds up into, because the vital thing is that shape determines function. So your 3 stages, stage 1 up here, the creation of that disposable messenger copy. Stage 2 above it, the creation of the polypeptide from that disposable copy. Stage 3 over there is the folding to make the specific shape so that protein can have a specific function. Okay, take a screenshot of this one if you want, because it's visually got the summary of what's going on. Now, obviously in the exam, you're not going to draw a picture. If it says explain protein synthesis, they'll expect you to write it out, and that could be a 6-mark question. There's normally 6 markers are the biggest markers that you get in the exam. So let's quickly whiz through the key bullet points, and I'll put the keywords in blueprint for you. So first off, let's give some background to the answer. The DNA, key thing, is made of nucleotides and holds the code for building proteins. Code is a key word because you've got to know that DNA is a code. Now the DNA is in the nucleus as structures called chromosomes. Important to state that because we know that being in the nucleus, the ribosomes can't actually see it. Important to know what chromosomes are. So an enzyme locally unzips the DNA strands to expose the bases. Remember I said it unzips, it separates them. And when I say locally, I mean local to the gene that you want. The DNA would be an incredibly long molecule. You only want the very local area for the gene, the bit of DNA that you're looking at. So you locally unzip the DNA to expose the bases so they can be seen. The enzyme then makes a messenger copy of the needed section of DNA, that's the gene. The messenger then leaves the nucleus and goes to a ribosome, and each amino acid is coded for by 3 bases which the ribosome reads. A specific transfer molecule brings the amino acid that corresponds to the 3 base codes. So they're like the little workers running back and forth to the ribosome. The ribosome collects this and moves on to the next 3 bases. Another specific transfer molecule brings the next amino acid, which the ribosome bonds to the first. Remember, that's a peptide bond. That's why it's a polypeptide. The ribosome then moves down the messenger code, bonding each new amino acid to the last. That builds the polypeptide of the specific sequence. Lastly, then, of course, that then folds in a very specific way based on its specific sequence, and the shape it folds into determines its function. Now, why that is so important, because by controlling that, by controlling protein synthesis, by controlling when different genes get expressed into their proteins, a cell can control exactly what processes it performs. If it wants to do one kind of process, it quickly expresses the genes into proteins. It does protein synthesis of the genes to do that process. Then those proteins will break down and disappear. So it stops doing that. And then it goes, right, well, now I want to do something else. So I'll get those genes and I'll make those proteins and I'll do that. So by a cell controlling which bit of DNA gets read at what time, it can control what proteins it makes and hence what processes it does. That's such a vital piece of biology. Take a screenshot of this. It's got the basic overview, but these 2 videos together are such a key, vital, important piece of the biology course. Okay, carrying on then in the genetics and inheritance playlist, I'd like you now to head to the meiosis video. Now, meiosis is the type of cell division that makes gametes, the sex cells. Within this video, I do a little recap of mitosis, which is the more usual type of cell division that just makes copies of cells. We did that way back in the cell biology playlist at the start of phase 1. So I will do a brief recap of it, and then at the end of going through meiosis, we'll look at a comparison between mitosis and meiosis, because quite often that's how they ask you about it in the exams. As always, I will ask that you please do subscribe to my channel on YouTube if you haven't done so already. It really helps me to have more subscribers on there, so I know what kind of things are working, what kind of thing is useful. Like this video if it was useful, and head over to my Facebook page if you want to find some more random biology facts. Thanks a lot.